What's possible in cancer treatment has been completely redefined in the last, I'd say, 5 to 10 years. It's really driven by the pace at which science has progressed in oncology, and we're now beginning to understand cancers at the molecular level.
More than probably any other field, cancer is leading the era of genomics, where we understand the basis of genes and its relationship to disease. First of all, we're able to target our medicines more specifically than ever before to offer increased efficacy and improved safety profiles at the basic mechanisms of cancer proliferation. Second of all, we're able to develop antibodies to specific proteins on cancer cells that deliver a very potent payload to kill the cancer. In addition, we're discovering gene signatures of patients' cancers so that we can match an individual's cancer with the appropriate therapy. And finally, we're now unlocking the clues to enlist the immune system to fight the foreign invading cancer cells.
One of the great mysteries in cancer therapy over certainly the entire time I've been engaged in cancer drug development over the last 25 years or so has been why our immune systems don't do a better job in their own right of fighting these tumors. It's only been in the last 3 to 5 years that we've really had some fundamental breakthroughs in that area that are now enabling us to develop this new family of immuno-oncology therapeutics.
Our purpose at Tesaro is to improve the lives of patients with cancer. And the way that we do that is to in-license, acquire, develop, and ultimately commercialize oncology and oncology supportive care products. For a company that's only 4 years old, we feel very fortunate to be preparing our first new drug application for submission to the FDA targeted for mid this year, and we're also preparing for commercial launch of that product next year.
Tesaro recently in-licensed 5 preclinical immuno-oncology programs. We're really excited about this addition to their portfolio for a number of reasons, primarily because we think that immuno-oncology has the potential to be a very broad background set of therapies that will be incorporated into many, many solid tumor and liquid tumor therapies over time, and will show synergy when combined with targeted therapeutics and other cancer-targeting therapies that Tesaro is already developing.
Immuno-oncology, simply put, is really directing the immune system to kill cancer cells. Under normal circumstances, the immune system will scour the body and identify tumor cells and eliminate them before we could even detect them with any tools that we have today. So a tumor, though, ultimately evolves a mechanism to shut down the immune system. Our goal in targeting the immune system for the treatment of tumors is to block the interaction between the tumor and the T cell, so the tumor can no longer shut off the T cells, and the T cells are then able to have their effect, which is killing the tumor. Immunotherapy has been kind of under the radar for many years, and it's just persistent, determined research that has grown in insistence over time until probably arguably at ASCO 2013. The clinical data that was presented there was profoundly changing. And even the most skeptical minds finally had to stand up and pay attention to the fact that this data was real. And it was really changing the way that we think about cancer care for patients. We really believe, and I think many in the field do, that immuno-oncology represents a sea change in the way that patients are ultimately going to be treated.
This could well become a foundational therapeutic regimen. And since combination therapy is the hallmark of approaching cancer, we envision immuno-oncology as a foundation and multiple different approaches combined with it in the future.
What we see at the FDA today is a real enthusiasm and embracing of targeted agents. They like to see the fact that you can select a specific patient population, put them into a clinical trial, and have an 80% response rate. That shortens the development time and the cost to develop these drugs and gives them much more confidence that when they approve a drug, it's really going to provide benefit to a patient.
What's really exciting right now is that we're actually having a debate about whether it's appropriate to use the cure word when talking about various cancers that just a couple of years ago were death sentences. The debate in the field now is around how far do you have to go? If we see sustained remissions beyond 5 years, is that a cure? Do you have to wait 10 years for it to be a cure? Do the patients have to be off all additional therapies to call it a cure? I just find it encouraging that we're actually having this debate.
In the past, oncology research was really targeted towards cancers of various organs. And in fact, you still hear people referring to cancers as lung cancer or breast cancer. What's become clear over the last 10 years since the war on cancer was declared and since the human genome was sequenced is that cancer is really a disease of the DNA, a disease of change within genetics.
Genetics is the study of variation at the level of the DNA sequence, but there's a whole apparatus of proteins responsible for taking a DNA sequence and expressing cellular proteins to provide a variety of functions. In cancer, this epigenetic system often goes awry, and it's very important to develop new therapies that address this abnormality in cancer cells. Epizyme is a company that has a whole platform of addressing epigenetic phenomena in cancer, and they're making great strides first in hematologic diseases.
For a 6-year-old company to have 2 programs that are in the clinic and 3 additional ones with our partner GSK moving forward aggressively towards the clinic is really setting a very high bar for drug discovery that we are very, very excited about. And it really is predicated on the productivity of our platform. Our platform is focused on a family of proteins called HMTs, and about 20 of these HMTs out of a total of 96 are potentially altered in driving either solid tumors or blood-borne cancers. One of the things that we're very excited about at Epizyme is the opportunity that this personalized therapeutic approach brings to patients and their families. Historical anti-cancer agents cause the kinds of side effects that we all dread: nausea, vomiting, loss of hair. But that's because they're general poisons. If we can treat the underlying genetic change, we hope that we can get not only greater efficacy, but also do it without those kinds of side effects. Therapies targeted towards genetically defined cancers are being very well received by the regulatory agencies as they maintain themselves at the cutting edge of oncology and cancer drug discovery. They understand that cancer really is a disease of altered genetics, and that epigenetics, the field that Epizyme studies, is really therefore aimed at one of these root causes of cancers.
The advances in science over the last several years that have driven our focus on targeted therapeutics in oncology and on immuno-oncology have to do with our new ability to understand disease at a molecular level, at a genetic level, and at a pathway level within the individual disease.
What I think we haven't appreciated deeply until about 5 years ago is that within each individual cancer, there's tremendous heterogeneity. There's more than one cell that's responsible for driving the growth and the proliferation of that cancer. There's more than one molecular alteration. Protein homeostasis is one of the basic pathways within a cancer cell that are required for survival. When you attack it, the results will be broad-based and there'll be long-lasting effects on the tumor, and more people will have their cure. I'm a scientist by training and have been in the drug development world since 1989. But in 2006, I was diagnosed with ovarian cancer. And we think, as we're in the lab working, that we know what cancer is about, and we know, the types of things we're going to do to change cancer. But when you become a patient and you really understand, all of the treatments, all of the treatment choices, all of the lack of treatment choices, the lack of really trying to individualize care, it really caused me to think about what do we need to do now to really change the paradigm for cancer treatment. Selecting the patients that will benefit from Cleave drugs is really at the core of what Cleave is about. In order to do this, not only have we had to identify the right targets, identify the right drugs that will hit the targets, but develop new methodologies and new analytical tools to decipher which tumors are over-dependent on protein degradation pathways for their survival. Cleave is at such an exciting time right now. It's exactly in a startup company what we live for. We've been working for 2.5 years on disrupting protein homeostasis, identifying the right targets, trying to find a lead drug that's good enough to take into the clinic, developing the right tools to analyze which tumors might benefit more. And that's all coming together for us now. This summer we will be starting our first clinical trials.
So it's really exciting the way science has completely transformed what's possible in terms of treating cancer today. Now that we're beginning to understand diseases at a molecular level and to be able to target fixing those diseases at the molecular mechanism instead of at the tissue of origin, we're really subsetting all diseases now. The models for how we develop drugs now, with parallel development of diagnostics, with biomarkers to enable early readouts, with patient selection, and with larger clinical benefit for smaller subsets of patients. That was pioneered in cancer drug development and rare and orphan diseases. But what we're now going to do, going forward over the next 5 to 10 years, is apply that to all disease areas.
This is a very special time in cancer research. And I've experienced what is a once-in-a-generation example of helping patients with cancer by developing new medicines. There's a lot more to do here, and NEA is very well positioned to harvest this important scientific information and translate it to clinical benefit for patients of the future.